Now, um, to give justice to those who have studied on the topic I assigned you, now we have to include this process replication for our semifinals. Okay? So, because we cannot understand fully the succeeding process if we cannot understand this topic. So you have to understand this topic first so you can understand succeeding topics. Okay? Now, let's have a review on the nucleic acids. Now you say nucleic acids, these are said to be one of the organic compounds, and they are made up of building blocks called nucleotides. Now these nucleotides are held together by phosphodiester bond. Now one example of nucleic acid is DNA. Another is RNA. Okay, of course there are some other nucleosides or nucleotides which are said to be included in this chapter, but we will not include them. So we will just focus on the role or the structure and the function of this DNA and the RNA. Now, let's have a review of its structure. Now, DNA is double-stranded. So this is one strand. So we have two types of strands for the DNA. We have the 5' 3' strand and the 3' 5' strand. So there are two strands. So how do you differentiate DNA from RNA? DNA is double-stranded while RNA is single-stranded. Now another difference is the sugar for DNA is deoxyribose. When you say D means removal, oxy, removal of oxygen. So the ribose minus the oxygen is already deoxyribose. And that is what DNA is composed of. So this is now the sugar. So you have the sugar, which is deoxyribose. But for the RNA, it is ribose. Now, so of course you have the phosphate bonded together with a sugar, and the sugar with a nucleic nitrogenous base, I should say. So this one is one nucleotide. This is another nucleotide because when you say nucleotide, it's composed of the phosphate group, the sugar, and the nitrogenous base. That is one nucleotide. And this is another nucleotide, so on and so forth. Okay? So we have this model actually. So this is the secondary structure of DNA, the helical structure, which is said to be two strands. And we have this red, the sugar, the white is the phosphate. So the phosphate bonded with the sugar with the base or nitrogenous base, meaning a base with nitrogen. Now, these nitrogenous bases are held together by a weak bond called hydrogen bond. Now, this hydrogen bond, so what binds these two strands together, 5', 3', and 3', 5' strands, is just actually a weak bond called hydrogen bond. So with the application of a small amount of heat, then this can easily be separated. So this can separate. Now, separation of these two strands is a requirement for the replication process, so as with transcription process. Because this has to be exposed. This nitrogenous bases sequence must be exposed. Why? Because this serves as the template or the model for the new strand to form. Okay? Now, what is this so-called, genetic information? What is this genetic information? Now, the sequence of these nitrogenous bases is said to be the genetic information. Why? Okay? Now, say for example, for every 3 nitrogenous bases, that corresponds to a particular codon. So one codon is composed of 3 nitrogenous bases. And every codon corresponds to a particular amino acid. And these amino acids are the building blocks of proteins, which are held together by a peptide bond. Get it? And that's how protein is made of. And how protein would be constructed or synthesized, that will be our topic. Okay? So as we know that protein is the most abundant organic compound in our body. So they could be in the form of enzymes, hormones, or structural proteins, or it could be transport proteins. So many types of proteins. And how are they synthesized? It is through this process that we'll be tackling this afternoon. Okay? So you have here the nitrogenous bases. Now, another difference for DNA from that of RNA is, of course, DNA has thymine, which is paired with adenine, and then cytosine paired with guanine, and vice versa. So there is no other pair. So adenine is always paired with thymine, and accordingly, the concentration of adenine is always equals with thymine in the DNA molecule. So whatever is the concentration of the thymine is the same with that of adenine because they are always paired. Okay. Now the same is true with cytosine. How much cytosine is present in particular molecule is also the same with guanine, because cytosine is paired with guanine and vice versa. Now, if we have 26 letters in the alphabet that makes up maybe a word or that construct a single word, and these words into sentence, and these sentences into a paragraph, and a paragraph into a story or stories, then stories into books, and books into library. Got it? Now the same is true with this. We have 4 nitrogenous bases, and out of these 4 nitrogenous bases, this can construct say codons, different codons, and codons into amino acids, and acids into protein, proteins, of course, to the whole individual. So we have this genetic library inside our body. Okay? So what comprises such library are those genetic informations, which come from the different sequences of these nitrogenous bases. Now, by the way, thymine is only present in DNA, and in RNA it has uracil. So uracil is only present in RNA, and thymine is in the DNA. Okay? So where you could find this DNA or RNA? Now, in a cell, say you have now the cell. If this is our cell, we have this nucleus. Now, we have these nuclear DNAs which are packed in a chromosomes. So where you can find that? It's in the nucleus. So you have here the nuclear chromosomes. Now, what are chromosomes composed of? Now, nuclear chromosomes are made up of DNAs and proteins. And these proteins are usually histones. Now, we have types of histones: histone 1, 2A, 2B, 3, and 4. Now, that organizes the DNA.